So we're in section 6, the last bit. We started this last time, but we didn't finish, so I went ahead and just threw it into this lecture. So it was all together. So you need to be able to describe the general structure of an IgG antibody. So the standard class of antibody. That was why attendance went slower than normal. Thank you. Fixed it. Okay. So general structure of IgG class antibody. So hopefully, right, you went over the different classes, and we're going to go over those today. So for an antibody, we talked about this last time, right? It's made up of 4 polypeptides that make up the overall protein. 2 of them are 2 identical heavy chains, labeled B here, and 2 of them are 2 identical light chains that the cell makes. And again, what cell makes these? That's a B cell, right? And what one actually secretes antibodies? Plasma cell. Selma's the only one that's going to ace this test. Oh, that's from my other class. It got mixed in. Yeah, there you go. Okay, so when we draw these little stick figures, we draw it kind of as a Y, right, shape. And again, it's kind of a stick figure for it. As you saw in the previous picture, and I talked about last time, there are disulfide bonds that connect the 2 strands, connect the different chains together. And then within the chains, and I'll rewind in a second, there are disulfide bonds. So notice here, those are disulfide bonds. So that's within a chain, but the really important ones are the ones that are intra-chain, which connect the 2 look, I hate these terms. ER, right, is between the chains. So a heavy chain binding to a heavy chain, and a heavy chain binding to a light chain. Intra, with an A, is within a chain itself. So these guys are intra, these are inter-chain disulfide bonds. The really important ones are the inter ones, because these are the ones that make the overall tertiary structure of this antibody, this protein. If the light chains didn't bind with the heavy chain and the 2 heavy chains didn't bind together, you wouldn't have a functional antibody, right? So how these guys function is what's important to us and what we're going to really focus in on today. So when scientists were studying this, they actually could break up this structure into 3 pieces. The 2 arms, known as the antigen binding fractions, abbreviated FAB, and they are pretty fabulous. And then the bottom portion, which actually crystallized in their fractions that they made. So that's where it got the C label. And it actually works out pretty good, though, because you end up with A, B, and C, right? On accident, not on purpose. So these 2 portions up here, which are identical, right? They're just mirror images of each other, circled here in orange. They contain the region that's variable, what makes each antibody produced by each individual B cell unique. Those variable regions I talked about last time, this is where an antibody differs, right? And that's where it actually binds to the antigen. So these 2 pieces they ended up with, right, we just said are what? The FAB fraction. And they contain the antigen binding site. So this part of the antibody they found actually would bind to antigens. Where this portion down here, which we know as the FC region, was actually the part that interacts with the immune system. right? So this is the part of the antibody, especially for an IgG, that phagocytic cells have a receptor for. This is the part of the antibody that they're grabbing onto, which makes sense because here at the other end is grabbing onto antigens. Does that make sense to you guys?
Is it because of the heavy chain?
Yes, right? So this is, good keen eye. Lance noticed this is part of the heavy chain, right? It's not all of it, because remember the heavy chains extend all the way up. So, but notice this is part of the constant region of the heavy chain, right? So this, for each class of antibody, this part stays constant, the same. If it changed, we'd be in trouble, right? Because then our phagocytic cells wouldn't be able to recognize it. So just as I talked about last time, that these constant regions are important because they allow, in this case, the two heavy chains to bind to each other, but the heavy chain to the light chain to have a functional antibody. Make sense? Okay. So the structure really determines how these things function. So in your DNA on chromosome number 2 and chromosome 22, there's the information to make two different types of light chains, what we call kappa and lambda. So they have lots of different segments of DNA that they can put together in different combinations to make the variable regions. Same is true for the heavy chain, because remember the heavy chain has a variable region as well. For them, they put together V, D, and J segments. For light chains, which are smaller, they just have V and J segments they put together. And then, of course, they have their constant regions. For the heavy chain, as Lance pointed out, part of their constant region makes up that FC portion and is the portion that interacts with the immune system. There are 5 different classes, although we're only going to focus in on 4 of them, of antibodies. These classes is determined by which portion of the DNA do they use to make which constant region for their heavy chains. So you'll notice that mu is written here. Mu is for M, right? In the Greek letters. That's for the first antibody that we produce.
Where is that?
M, right here, mu. Mu. So that one's first because that's what we always make first. And then the B cells can switch. They can switch to the other classes, and they abbreviated him here together, right? So gamma for G, alpha for A, and what they're not showing here is E, epsilon. They use the Greek letters for it. And this is D, delta, which we're not going to talk about. So class switching, they're going to switch out, as I showed you in that diagram, which C region they're going to make. And this leads to the different class of antibodies that we're going to talk about. So some of them have slightly different structures than the IgG that we just went over. They're a little bit more complex. Especially IgM, which I said is the first class of antibody you're always going to produce. And then it's going to be signaling from guess who that's going to help the B cell determine which class it's going to switch to from M. Who talks to B cells and tells them stuff? Who talks to B cells? And B cells are antigen presenting cells, and they actually present to these cells. What cells do B cells present antigen to? Via MHC class what? 2, right? Who do they talk to? T helper cells. Which subset even? 2. So now we're going to look at the different classes, right? Like I said, we're going to look at 4 out of the 5. So you should probably have this labeled, right? So don't look at it. Flip it over. I want to see how much you remember. Okay, so this one is called secretory antibody sometimes because you secrete it in, notice, your mucus, covering all your mucous membranes, your tears, your breast milk, and your saliva all contain IgA, right? So I always remember this is the most abundant one you make, by the way. You make tons of this stuff, right? Because you're always making mucus and secreting it, right? Of course, we only produce breast milk at a particular time, but we produce it. Why would we want to put antibodies in breast milk? Give it to the baby, right? To help protect the baby. It's passive immunity. It's past immunity, right? Did they make those antibodies? No, but will they work for them? Absolutely. Absolutely. The bad thing is that they didn't make them, they don't have what? Because remember when we have memory cells, right? So it isn't until the baby's exposed, right, and produces their own antibodies will they have memory of it. But you can pass immunity on to them passively in the form of breast milk. And that's why nowadays, too, people are going back to breastfeeding more than they did in the past because we can't replicate that benefit. We can't put IgA in formula, not cheaply anyways, and not effectively, right? So that's an immunological protection that only you can give to your baby through natural breastfeeding. So this one actually, we saw IgG, that structure we went over is sometimes referred to as the monomer, right? The single unit that's produced. But two units can be put together to form what's called here as a dimer, right? So di meaning two. So the IgA that's produced by the B cell, two of them get hooked together, right, via another protein. You see zigzagging here, this pink one, hooks two of them together.
That's a protein?
Yeah, it's a protein. It's all protein. And this helps get it to be secreted. This is kind of a signaling thing. Oh, okay, this goes into mucus, this goes into tears, right? This goes into our saliva. So as we're going to see, what are the things that antibodies can do? One of the things that we'll look at is agglutination. Actually be able to bind to multiple antigens at the same time and clump them up. The other thing is that the FC portion, although it's kind of hidden here, can be grabbed by phagocytes. So IgA can be in its monomer form in our blood too and throughout our body and can be picked up by phagocytic cells. But remember, if we secrete it, if we add this protein to it and we secrete it into our breast milk, into our mucus, into our saliva, is that in our body? No, secreting means it's leaving our body, right? That stuff is leaving our body. Phagocytic cells don't leave our body, y'all. They stay inside. They're not going to bind to it in this dimer form. They're going to bind to it in its monomer form. Does that make sense to you guys? Okay. So moving on to this next one, when it's attached to antigens, or in fact, it can attach to the surface of basophils and mast cells. So when antigens aren't even there, this one can bind to mast cells and basophils. And then when the antigen comes along, those cells are going to degranulate. They're going to release the substances inside their granules, right? In the case of mast cells, they're going to release things like histamine. So remember, this is the one I don't like. Because this is basically an allergic reaction, right? So this is IgE for Erica, right? You won't ever forget it. And so specifically this FC portion, this bottom portion, right? Those cells have a receptor for it. Basophils and mast cells have a receptor for it. Now, they put eosinophils in here, and you really need to cross them out.
What?
Eosinophils.
Yeah, in the beginning?
Yeah, eosinophils don't bind to this class of antibody. They bind to a different class of antibody, and they do the same thing as mast cells and basophils do. They degranulate. But it's not in response to IgE. It's a different class. So this monomer, right, is the one we've been going over. This is the major antibody throughout your body. And the good or bad thing, depending on how you look at it, this one can cross the placenta. And this one, as you can see, has the most actions, right? It can activate complement. It can opsonize cells for phagocytosis. It can help neutralize toxins. It can participate in antibody-dependent cellular cytotoxicity by natural killer cells. So this is our major worker antibody. And which one is this? This is IgG. And then the last one is actually the very first one we make in response to an antigen. It's pretty much mostly just present in our blood. One of the things they don't tell you here is that this one's too large to cross the placenta. This one does not cross the placenta. And in fact, the only one that does is IgG. But this one, look at how big it is. When it's secreted from the B cell, they stick 5 monomers together. That's why it's called a pentamer. Pent meaning 5. So remember, each antibody has 2 antigen binding sites, so this one has a total of 10 antigen binding sites. So this one can bind to a lot of antigen. And this one is IgM. And I always remember mega big, right? M for mega big. This guy is huge. So he's really good at agglutination, right? Because he can bind to a lot of antigens. And he's also the other one that can activate complement. So they talk about hemolytic disease of the newborn, right? This is where if you are an Rh negative mother, which means you don't have that antigen on the surface of your red blood cells, you could potentially have a baby, though, that is Rh positive. If you get exposed to, and the good news for this one is you've got to be exposed to it, and so you can somewhat avoid that. You've got to be exposed to positive blood. If you're exposed to Rh positive blood, you will probably make antibodies against it. The class of antibody you make, unfortunately, is IgG. So what can that do if you have been exposed previously by a previous pregnancy where you didn't receive treatment so that you wouldn't make the antibodies, or some idiot gave you a blood transfusion of the wrong type of blood, right? If you somehow got exposed to positive blood and you are a negative person, right, Rh negative, you could make antibodies. Why do positive moms not have nothing to worry about? Are you going to make antibodies against yourself? No, not unless you're experiencing autoimmune disease. So Rh positive moms don't have to worry about this because they should never make this antibody, right? Because they have that antigen. You're not going to make antigens against yourself. But if you're Rh negative and you get exposed to positive, you could make these antibodies, right? Which means that if you're pregnant with a positive baby, those antibodies do what? They cross the placenta and they can attack the baby and cause a disease known as hemolytic disease of the newborn. Hemolytic meaning that, guess what? When those antibodies bind, all of these things can happen. Complement can be activated. Natural killer cells can be activated. Opsonization by phagocytic cells. And destruction by phagocytic cells. All those things can happen to the baby's red blood cells because your IgG crossed the placenta and started attacking that baby's, started labeling those baby's red blood cells as foreign.
There would have to be a transfusion.
Not necessarily. I'm not going to get into the details of it in this class, but I just want to make you guys aware of it. We don't have to worry about the ABO blood group because guess what class of antibodies you typically make? Just IgM. That's why you don't have to worry about that part of your blood type that we talk about. You make a different class of antibody. Okay, so let's see who was paying attention. when we get to the outcomes part, because I'm going to ask you again, what ones can the different ones do? Okay, so class switching we just talked about, right? Where with the help of the B cells, they're going to switch to these different ones that we talked about. So they're going to start out making IgM, but they may switch to E depending on what we're dealing with. They may switch to A if we want to secrete it, right? And they may switch to G, which is the most common switch. In addition to switching, during the response, they're going to make changes. They're going to cause mutation in the variable region of their antibody. So they've been activated, right? Their antibody binds to an antigen. Now they're going to change it. Why would you want to do that? Why would you change something that's working? Well, why do you practice any sport? To get better. Well, if you change, is there a possibility you're going to get better? Absolutely, right? And so they're going to modify. And so the question they ask you is, how is it different for primary and memory response? So remember, a primary response means what cells are being activated. If this is the very first time you've ever seen this antigen, naive cells. So we're not only are we maybe waiting 7 to 14 days before they even find ones that match and we start producing antibodies. So we're going to start out with IgM and then maybe we'll class switch. And this is going to take time, right? Where if we've already seen it and we're activating memory cells, some of those memory cells have already class switched, right? We can skip over M, go right straight to G or A or E. That decision's already been made previously in the first exposure. So that's the difference with primary, right? It's going to take time for class switching to happen. Where if it's a memory response, if those are memory cells, they may already be class switched. They may already be memory IgGs or memory IgAs. Programmed to go that route. The other thing too is they may have gone through this process already, that memory cell has a really good affinity for the antigen. Affinity, we talked about this last time. What does this mean? What does affinity mean? How well that antibody binds to the antigen, how strong that binding is, right? We want the best possible, strongest binding, right? We want the closest association, the best grip, right? So the best pair of gloves we can buy, right, to grip onto something. No, this is in the variable region. Right? So affinity maturation is where they change their variable region in hopes that their binding is more specific, right? Better affinity. And this happens in the primary response, but think about this. As they're going through this response, the B cells, as they're going through clonal expansion, and they're making these changes as they go. So unlike T cells where they just pump out all the exact same copies, right? B cells do more stuff. They class switch. They mutate their variable regions in hopes of greater affinity for their antigen. And they're going to produce memory cells each time they change. So here, this is a diagram I kleptoed from someplace else. It's not in your book. Okay. And so the pluses here on the B cell indicate how strong the binding is. So this is a good binding. This is a 2+ binding. And then they go through mutations. Some of the mutations actually result in not as much binding. So notice they're not going to get a strong signal to proliferate, right? That lineage is pretty much going to die off. This lineage, right, is going to continue. That's the 2+. Say it goes through another round of mutations. And in this case, it ends up with a mutation that has a much stronger binding. We've got a 3+ here. That's really good. So they're going to get the strongest signal to continue, right? And the population of B cells that we have that are then going to differentiate into plasma cells and memory cells are going to have the strongest binding to the antigen. Right? So we kind of tweak it, right, as they go. This is different from what T cells do. So affinity maturation happens, right? And so in our second response, this is why we're so much better, right? Because we've already tweaked it the first time, right? We've already got the best. This is another diagram that I kleptoed, which is a good one. Yeah, this is in the new one that I didn't post for you guys because it has all the answers to what we're going over today. I don't want to give it to you ahead of time. Okay, so in the DNA, remember this is what chain, heavy or light, is going to determine which class of antibody. Is the heavy chain, right? The longer one that has the FC region. So notice here they've got them labeled the classic letters, right? So here's all the 5 different classes of antibodies. We didn't talk about D. We talked about the major functional classes. The very first one we're always going to produce, because notice it's first in the DNA, is IgM. And that's actually the class of antibody that's on the surface of your B cells as a B cell receptor. When it's stimulated by a helper T cell, it may switch to, say, G. In that case, y'all, you know what it does? It deletes the ones before it. So once a cell switches to that, and you have memory cells produced to that, they can only produce G because they've switched. And of course, other switches they could choose is to cut out all of this and be E, and they can cut out all of this and be A. Yeah. So, I mean, they literally change their DNA, though, when they make this switch. So this is a permanent switch. You can't go, oh, I want to go back to M. No, no, no. Doesn't work that way. They get rid of it. They throw it out. So the really cool stuff. And we've already seen most of the show. Most of these are review, right? Because we've talked about what we're born with, our innate immune response, right? And remember, these aren't separate. The distinctive thing with the adaptive is that we develop memory, right? But our antibodies that we produce can interact with components of our innate immune response, right? What we're born with. So this enhances our innate response, right? Having antibodies, this product of an adaptive immune system, right? Which not all organisms have, right? Just invertebrates like ourselves. So I listed them in the simplest of terms here, right? And this is exactly from the labeling packet I gave you. So we're going to go through each one of the pictures and we're going to add the label to it, right? So which one of these pictures is neutralization? Which is complement fixation and lysis? Which is opsonization? Which is agglutination, which I've mentioned, right? When we talked about some of these antibody classes, I said they're really good at agglutination. But let's look at what that actually is. Activation of eosinophils and basophils and mast cells, and I'll make that distinction in the different class of antibodies there. And we've already seen this, you guys. Antibody-dependent cell-mediated cytotoxicity. What cells, what white blood cells specifically will bind to antibody-coded cells and kill them because of it? Natural killer cells, right? So we talked about this when we talked about natural killer cells. We're going to add in one more level of detail to that. What antibody class can actually do this, right? All right, so for this first one, right, what is this? So this is a cell. Looks like it's infected by a virus, right? It says viruses. It's got virus proteins on its surface. We made antibodies against it. They're binding. This guy, which is who? Natural killer cell, recognizes this antibody, right, by this portion of it sticking out. And that induces this cell, this natural killer cell, to kill this infected cell, or to get it to go through apoptosis by releasing perforin and granzyme. So programmed cell death. So what is this process? This is, this is antibody-dependent cell-mediated cytotoxicity. What antibody class causes this? It was only one of them. IgG. She's on it. IgG is the only one. So that receptor on the natural killer cell, it's actually specific for that class of antibody, which is our most abundant ones in our body. Right? Inside of our body, it's the most abundant. So for this next one, right, we've got, say, some bacteria potentially. It's antigens on its surface, and we've made antibodies against it, and they're binding to it. And so macrophages and neutrophils have receptors on their surface for the Fc portion of our antibodies. So this is going to cause them to engulf and kill it. But we're basically tagging them for destruction, aren't we? And what do we call that process where we use some molecule to tag them for destruction by phagocytic cells like macrophages and neutrophils? Opsonization. So I didn't put this on the slide. I wanted to, and I didn't come back to it. Complement, right? That series of proteins. There's one of those protein fragments that's a really good opsonin that will help tag it for destruction by these phagocytic cells. What complement fraction is that? C3b. Remember B for binding, right? C3b. That's a really important opsonin other than antibodies, right? Antibodies are really good opsonins because our phagocytic cells have receptors for them, right? And they know if it's got antibody on it, or C3b, it needs to be destroyed. Right? So opsonization, remember, think tagging for destruction. So we've seen this before, right? So we talked about how we can tag them for destruction. So here, I covered up the cell with my picture label. So this cell is a mast cell or a basophil. It has receptors on its surface for IgE. So what this picture is showing you is activation of basophils and mast cells. What they're not showing you that would have a similar reaction is eosinophil activation. So what class did we say activates basophils and mast cells? The one shown here, right? IgE. And this is, you know, people who are allergic have been sensitized and produce IgEs, but also against multicellular organisms that we can't really engulf, right? So we're going to have to throw toxic substances on them. So parasitic worms and such like that, right? We usually have an IgE type response to. You get a little bit different response to eosinophil activation, and therefore it makes sense that there's actually a different antibody that interacts with eosinophils. Anyone want to gander a guess? Can't be, it's probably not A, right, because we secrete that. We already used E. We've got M and G left, right? So which one? It's G. It's IgG. It's the one that actually interacts with eosinophils. What are they showing in this picture? IgG, yep. Eosinophils, it's IgG. So what's C1? What's a MAC? That's what they're showing right here. This is C1 and this is a MAC. So what's happening here? What system are we activating? Because an antibody bound to a cell. Which one has C1 and the MAC? Complement, right? This is activating complement via the classical pathway, which can lead to the formation of the MAC complex, which, what does MAC stand for?
It's membrane.
Yep. Membrane is the important part of that. Attack complex. Membrane attack complex. So that's this complex of proteins of the complement system that actually insert into the membrane and actually cause what to happen to that cell? We lyse those cells, right?
Does it destroy it?
Yeah, it destroys it. It lyses it. So who did we say was more susceptible, gram-positive or gram-negative to this, and why? Gram-negative, because they have what structure that this can insert into? Remember, this is why I make you know what MAC stands for. It is a membrane attack complex. The outer membrane of gram-negatives is what's vulnerable to this, right? Where the gram-positives have that peptidoglycan cell wall protecting them. Where the gram-negatives have that outer membrane. That's the outermost covering. It's exposed, it's vulnerable to attack by the MAC complex.
Which is why
Lysozyme attacks what? But what in the bacteria? Peptidoglycan, which is what makes up their cell wall, right? So in that case, who is more susceptible? Gram-positive or gram-negative? Gram-positive. So see the 1-2 punch? Complement, for the most part, is going to take care of gram-negatives. Lysozyme, that enzyme, is going to help take care of the gram-positives. Good one. Somebody's thinking. So when we were going over the classes of antibodies, there was actually 2 of them we said could activate complement. Which 2? Think about it. Which ones do we want to be able to do? What's the first antibody we produce all the time?
Can you say the first one again?
Which ones actually can activate complement? There's only 2 antibodies that can do it. IgM and IgG. Remember, the first one we produce, IgM. So the first one we produce, we want to be able to activate complement. And the most predominant one in our bodies is IgG. Right? So this creates that link, as I was talking about, of adaptive to innate. Right? This is an innate response complement. Right? This series of proteins. But we can help our response by producing antibodies. Whew. We only got a couple left, thank goodness. Okay. So in this one, you see a virus, right? Can someone close the door? Because I have a feeling other classes are going to let out and it's going to get really noisy. Yep, see? I knew it. I could tell. So we have a virus here, right? An antibody is binding to it. Remember, the first thing that a virus must do before it can infect our cells, it has to do what? It has to bind to one of our receptors. If there's an antibody binding to it first,
Is that agglutination?
nope, it's not agglutination. So it's binding to it first, and so therefore it can't bind to us. So this is said to be neutralization. Now they're showing a virus here. But this can be true for any of the attachments that needs to happen, right? If a bacteria needs to attach to us, right? We can stop that attachment by antibodies binding to it. Toxins, usually to affect our cells, guess what they've got to do? They got to bind to them. If they're coated in antibodies, can they bind to us? No, and that's really where this name neutralization comes from, because you can neutralize toxins by having antibodies bind to them. right? So toxins, pathogens like viruses and bacteria that need to get inside, if we coat them with antibody, we can stop them from attaching to our cells and essentially neutralize them. Right? So that's a really awesome protection, right? Of being able to block them from getting in.
This would be like for a person who would be immune to something but still can carry the virus in it. That could be a carrier.
That's a complicated one I don't want to get into.
All right.
I don't want to confuse other people.
No worries.
I'll answer for you later if you want.
All right.
We're staying general. Okay, so this is one of the ones that we really haven't talked about yet. So this is where you'll notice that the virus, virus, they got it labeled virus. Because antibodies at the very least have 2 antigen binding sites, notice that this antibody is grabbing onto 2 viruses at the same time. And this one is grabbing onto 2, and so then they start clumping up, and this reaction is referred to as agglutination. And so when you guys take anatomy and physiology too, right, and you do blood typing outside of the body, you can actually cause an agglutination reaction to happen too. All you got to do is put an antigen and antibody together, and guess what? They bind to each other. This will happen even outside of our bodies. And so you can see that reaction, what we call an agglutination reaction. If you have red blood cells and you put a known antibody, like if you don't know what your red blood cells are, you put them on a slide or in a well, and you add known antibodies, say the antibody against the A antigen, and you stick it into that well, they're going to agglutinate, they're going to clump up like this. So you can see this without even using a microscope, right? They create a big clump. Why would this be beneficial, though, inside your body? Why would we want to clump them all up? You know, if we clump a whole bunch of them together, then a phagocytic cell like this one here can come along and very easily grab a whole bunch at once instead of just one. Look at how many he's getting to eat there, 4. But we could put more than that, right? So what classes of antibodies do you think are good at this? At clumping them up? Pretty much all of them, right? There's one that's like a super grabber, though, because he's so huge. IgM, right? And that's the one, remember I said you make against the ABO blood group? What's this one, though? This is IgA, right, that we secrete. And again, it has how many antigen binding sites? 2 here and 2 here, 4. So this is really one to agglutinate outside of our body, right, in mucus. It's really good at trapping bacteria, multiple ones, right? But you're right, the super guy is IgM. Because look, he can bind, he has 10 antigen binding sites. So in this one, look, one molecule is binding 5 red blood cells in this picture. That's why when you add that IgG against A antigens, it really clumps up. The same thing happens for B. If you have that antigen, you get a really good agglutination reaction. You get a little bit different, not as strong agglutination against RH, because what antibody did I say we make against RH? IgG. And remember, that one's just a monomer, right? Can only bind to 2 antigens at the same time. So this is another one I kleptoed from another book for you guys, because I like it because it kind of summarizes them all on here. And it really kind of almost adds in an extra one that we didn't talk about. But a lot of times you could just combine these two, right? Because as I said, if antibodies bind, they can really interfere with the ability of a pathogen of interacting with us, right? So in this case, again, they're talking about stopping them from adhering. But think about this too. If it's a bacteria that has flagella and antibodies bind to it, what else could we do? So when I was doing research in Bayou Trepanier, we're going down the bayou, right, in a boat, and all of a sudden it stops. We're like, what the heck? Pull the motor up, wrapped around the propeller is a sweatshirt. I was like, okay, that's why we're not going anywhere. Tore the sweatshirt off the propellers, stuck it back in, and down the bayou we went. So think about the bacteria, right? Their flagella is their propeller, right? What if we put a whole bunch of antibodies on it? It's like putting a sweatshirt on it, right? And bunches it up. So guess what they're not going to do anymore? They're not going to swim anymore. They can't swim through our mucus, right? If we attach a whole bunch of antibodies to them. We're going to clog them up and stop them from moving. That's what they refer to as immobilization. Yeah, and so if we can slow them down, clump them up, then our phagocytic cells are going to come along because the antibody is also an opsonine, and they're going to digest and kill them, right? We may even activate complement and lyse them that way, right? Or if it's IgG, natural killer cells might come along and attack that cell, right?
Can this one save then?
I'm going to post it right now. So that's it, y'all. It's a beautiful, complex world that we live in. And believe it or not, we just scratched the surface.
Test is on Monday, right?
Test is on Monday in the Hibernia Center. And as I said earlier, but it wasn't showing up on the screen, there is a lovely announcement, right, that lists it all out for you.
The whole,
Thing, yep. And so you'll notice there's one more bonus opportunity, right? There's a talk on Zika virus that's happening here on campus on Monday the 28th in the afternoon. If you go to that and submit your notes, you can get up to 10 bonus points.
I'm gonna need
I
Okay.
have a question.
All right.
there you go.
This part.
Mm-hmm.
Why do you
Now what?
explain the version for me, please?
So what they're doing is they're releasing enzymes, so perforin and granzyme, and what these do is cause that cell to go into programmed cell death, known as apoptosis. So that virus-infected cell, the reason why that natural killer cell is doing that is because it's recognizing the antibodies attached. So it's really the antibodies that are specifically recognizing that cell.
Oh, okay.
Without the antibodies, you wouldn't get
Yeah.
that response.
Yeah.
That's why it's antibody-dependent cellular cytotoxicity. Okay, so let me post it. Let me end